kind of mentioned the juxtaglomerular complex in passing when I told you that it's important that the distal convoluted tubule passes directly between the afferent and efferent arteriole. So right in that area where the DCT passes between the two arterioles, we've got a couple different types of cells that kind of work together and form this juxtaglomerular complex. This is an endocrine structure, meaning that it releases hormones. It also releases some enzymes that are related to hormones. But the whole point is that it releases a lot of stuff that's involved in controlling blood volume and ultimately how much we filter in and out of the glomerulus. So when we look at the juxtaglomerular complex, we see that it releases erythropoietin. Remember, erythropoietin is released when we don't have enough oxygen or blood coming to the kidney. Right? And the thought there is if we don't have enough oxygen being delivered, maybe we need more red blood cells. Maybe that'll fix it. So what does erythropoietin stimulate? Erythropoiesis, which you said is red blood cell production. Okay, we talked about that quite a bit in the first chapter that we did. The juxtaglomerular complex also releases renin. And renin is an enzyme. And what's renin related to? Like what big system? What's up? RAS, right? So renin, angiotensin, aldosterone. Okay, and what does that do overall to say blood pressure, blood volume? Increases it, right? Remember when to give like for blood pressure medicines, we block this. So this increases blood pressure. Renin, angiotensin, aldosterone system. We'll talk about that in a lot of detail in multiple slides coming up. Also, the juxtaglomerular complex releases something called adenosine. Adenosine's a paracrine factor, not an endocrine factor. Remember, paracrine factors just they travel through the interstitial fluid, right? And they just, tell things what to do in that little area. But what adenosine does is it causes afferent arterial constriction. So it says this arteriole that's bringing blood into the glomerulus, it makes it smaller. So less blood comes in, there's less pressure, there's less filtration. Okay, so you have less glomerular filtration occurring. Again, we'll talk through all of these as we go. The whole point is this juxtaglomerular complex influences how much we filter out of the nephrons. Right? Either increases it or decreases it depending on what's going on in the body. The complex right here is formed by cells of what we call the macula densa, and then also juxtaglomerular cells. So the macula densa refers to the epithelial cells of the DCT. That's what I drew right up here. So this is the afferent arteriole, right? That's the efferent arteriole. And this distal convoluted tubule, we said, passes between the two. So when we look at the cells that line the distal convoluted tubule, the ones that are right here, right up against the renal corpuscle, we see that the cells end up being like really kind of tall and stretched out and really densely clustered together. So you see like nucleus, 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 really, really densely clustered. So we call that area right there the macula densa. Then we see that over here, in conjunction with the smooth muscle cells that line this afferent arteriole, right, this is the one that brings blood in, those cells are called juxtaglomerular cells. And then this whole area together, the cells of the macula densa, and then these juxtaglomerular cells around the afferent arteriole, those together are called the juxtaglomerular complex. Again, different parts release different things, but everything's related to controlling glomerular filtration. Okay, here you see that. The afferent arteriole coming in, like the juxtaglomerular cells are the cells that are like the smooth muscle cells that are lining this right here, right by the glomerulus. This is the distal, nice. This is the distal convoluted tubule. Again, right by the glomerulus, these cells, you can see how they're skinnier. They're like squished and stretched out, so the nuclei get really, really densely clustered together. That's what we call the macula densa. And that area in general is important for maintaining glomerular filtration, right? How much we actually filter out of the glomerulus. All right, so the juxtaglomerular complex is important for maintaining homeostatic GFR. What is GFR? Glomerular filtration rate. Okay, how much we actually filter out of the glomerulus. And this is really, really important to make sure that we have a healthy or kind of immediate GFR. So we have to filter enough out to get rid of wastes, right? Remember, metabolism generates wastes all the time. So we make urea, we make creatinine, we make these waste products constantly throughout our body, and we cannot let them build up. They have to get filtered out of the kidney, like no matter what. Remember, a quarter of our blood every minute goes to the kidneys. So we've got to be cleaning it out all the time. We have to have GFR high enough to clean the blood. Now, we don't want GFR to be too high. If the glomerular filtration rate is too high, that means there's too much pressure, right? And we're pushing across that nice, delicate little membrane with a ton of pressure. And what happens if you have a filter and you blow it out with a ton of pressure? You damage the filter, right? You start to break the filter. So we don't want GFR to be so high or pressure to be so high that we damage the kidneys. So we want to make sure that our glomerular filtration rate is just right. And it's going to change a little bit, right? As blood flow and blood pressure change, it changes. But we do whatever we can to keep it right in a nice homeostatic range. And we can adjust glomerular filtration rate via a few different things. We can do it with adenosine, which we'll talk about in a second. We've got the renin, angiotensin, aldosterone system. And then we have different hormones like the natriuretic peptides. So adenosine, again, we said is a paracrine factor. So it's released right from that juxtaglomerular complex, and it just travels through the interstitial fluid and works right in that area. Okay, so it's not traveling all over the body, it's just a little local paracrine factor. And adenosine gets released when glomerular filtration is too high. Again, that means there's too much pressure and we're damaging that filtration membrane. We're damaging those capillaries. So the afferent arteriole goes in, efferent comes out, and we've got all these like nice little intertwined fenestrated capillaries here, right? So when GFR is too high, we've got a ton of blood volume and a ton of blood pressure pushing in here. And that stretches this afferent arteriole, right? If you've got a ton of blood being crammed in here, that stretches it. That tells it that it's dangerous, right? This is too high, we shouldn't be stretching this much. So we release adenosine, and what adenosine does is it causes contraction of this afferent arteriole. So now instead of being, say, this big, it's only this big. So now you have less blood going in. Less blood volume means less pressure, means less damage to the filtration membrane. Does that make sense? Okay, nice little local response. Renin is released from the juxtaglomerular complex in response to the opposite. So when we have a decrease in the glomerular filtration rate, or GFR, remember, this is not okay. We have to filter enough blood to get the toxins and wastes out. So your blood pressure might go down. You might be sleeping, resting, blood pressure is really, really low, but that doesn't mean that you have to stop filtering your blood. Right? You still have to filter the toxins and wastes out of your blood. So we have to maintain our glomerular filtration rate even when blood pressure drops. Renin is one of the things we use to do that. So remember this whole kind of conversion. Angiotensinogen, remember, is this inactive right? It's just a protein that's normally in your bloodstream. We make it in the liver and it just floats around in your bloodstream inactive, okay? Renin converts, so we use renin, that's the enzyme that converts angiotensinogen to angiotensin 1. Then another enzyme, ACE, which remember stands for angiotensin converting enzyme. Converts angiotensin 1 into angiotensin 2. That happens in your lungs and in your kidneys. And we have this ACE enzyme. That's where we've got ACE. And that converts angiotensin 1 to angiotensin 2. Angiotensin 2 is an extremely potent, extremely active compound. Angiotensin 2 causes all sorts. Remember, it causes, we'll talk about this later, but the release of antidiuretic hormone, the release of aldosterone, which both increase blood volume. It causes systemic vasoconstriction, which remember increases blood pressure greatly. Lots and lots of effects. Thirst, it causes lots all over the body. But overall, what angiotensin 2 is going to lead to is an increased blood volume and pressure. Remember, the whole point, the whole reason we did this was because we had a decrease in GFR. We're not filtering enough blood out of the kidneys, right? Not enough blood is getting filtered out to get the toxins out. So we have to increase that glomerular filtration. How do we do it? More blood, more pressure. So we increase blood volume, we increase blood pressure. Now there's a lot more force pushing out, and we increase how much we're filtering out of the kidneys. Does that make sense? Okay. It's an odd place to stop. Oh, shoot. Is everybody okay with kind of the overall process, right? We filter out of the blood and into the capsule. We maintain that glomerular filtration because of what's happening from the juxtaglomerular complex. After we filter stuff out, it goes through the tubule. We reabsorb what we need out of the tubule. Different things get reabsorbed in different areas of the tubule, and then we end up with urine. Yes?
So for renin and angiotensin, are they continuously released back and forth to maintain the right GFR, or do they only get activated and then stop once it gets back to the rate?
Renin and angio, or you mean renin and adenosine? So we'll talk about the different levels of control towards the end of this lecture, but they're actually, we talk about them together then because they both come from the juxtaglomerular complex, but they're actually different levels of control. Like the renin gets released because of sympathetic stimulation, and the adenosine is just strictly local control. So it's much more of a small little quick happen and then stop, happen and stop, happen and stop, whereas the RAS is like all over the body, longer lasting. So yeah, it's kind of odd to talk about them like they're equivalent because they're really not equivalent. The renin-angiotensin is a much bigger response than adenosine, longer lasting. All right, so we have to mention transport maximum and renal threshold. When I first start to talk through this, it's gonna sound confusing. I promise it's not. Like just walk through it with me and it makes sense. So these two things, these are different values. The transport maximum is a different thing than the renal threshold, but they're directly related to each other. Okay, we see that the transport maximum ends up determining what the renal threshold is. So remember that we should reabsorb all of the nutrients that get filtered out, right? We've got, I should just leave this. We've got our glomerulus, right? I'm gonna draw it as just one circle. We have our glomerulus. We filter out tons of nutrients just based on their size. and then we have to reabsorb them. So as this fluid goes through the proximal convoluted tubule, we have pumps, right? These active pumps. And what these pumps or transporters do is grab a hold of the nutrient and pump it back into the body, right? To reabsorb it. Now you see that we're reabsorbing things based on these transporters, right? Like they have to be there. This diffusion isn't just happening through the cells. These are transporters. They're grabbing the nutrient and actively reabsorbing it back into the body. So what we see happening is, if say the blood concentration of something increased, like glucose. Okay, I eat a meal, I've got a ton of glucose in my blood. That means that when I filter the blood out here, I'm gonna have a lot more glucose in this fluid, right? I had a ton of glucose in the blood, that means I'm gonna have a ton of glucose out here in this tubular fluid. Now, if that happens, that means I have to reabsorb more glucose, right? If I have to reabsorb 100% of my nutrients, the more there are, the harder my transporters have to work, and the more I'm going to have to reabsorb. Now, this happens until a certain point, and we call that Tmax. That happens until all of the carrier proteins get saturated. So say I have 1, 2, 3, 4, 5, 6 carrier proteins. And here I have 1, 2, 3 glucose molecules. That's fine. I can reabsorb everything. Then I eat a huge meal, right? And now I've got a ton of glucose in my blood. So when I filter this fluid out, I've got tons and tons and tons of glucose in it, right? So now all of a sudden, every one of these carriers is working. They're all pumping at full steam, but they're not able to reabsorb everything, right? They're all saturated, yet I still have glucose making its way down in the tubular fluid. Okay, I've met or exceeded my Tmax. Transport maximum is when all of your carrier proteins are saturated. That's the most that you're able to reabsorb back into the body at any given time. Whatever that is, whatever your essentially like your number of transporters are, that determines what the renal threshold is going to be. Now, the renal threshold is an actual serum concentration or blood concentration, right? Because we can't very easily go to somebody and measure how many transporters they have in all of their nephrons, right? Like that's just not feasible. But it's really easy to poke someone and see what the blood concentration is, right? And I can see what your blood concentration is, then I can look at your urine, say is it in your urine or not. Then I can give you more sugar, check your blood concentration, and then check your urine. Is it in your urine or not? Like those are easier things to monitor. So we monitor the renal threshold. The renal threshold, again, is just the blood concentration of whatever nutrient that correlates with the transport maximum. If we go higher than that renal threshold, any blood concentration that gives us enough nutrient in that tubular fluid to go over that transport maximum means that we're going to exceed or go over the reabsorption capabilities of the kidney. And then where do you think that nutrient's going to end up? In the urine, right? And it shouldn't be there, we said. Glucose shouldn't be in your urine. Under normal circumstances, amino acids won't be unless you just eat a bunch of meat. The only reason they'll show up in the urine is if we simply cannot reabsorb that much because we're using all of our transporters at max. So we said that renal threshold is a plasma concentration or a blood concentration above which that specific nutrient begins to appear in the urine. Now, this value is different for different things, right? It's not like we have one transporter that moves everything, or we have the exact same number of transporters for glucose and amino acids. They're different. So we have different renal thresholds for different substances. We'll look at glucose and amino acids. So the renal threshold for glucose is about 180 to 220. So what does this mean? 180 milligrams per deciliter. What's that like? Is that in the urine? Is that in the tubule? What fluid? The blood, right? This is the blood concentration that we're talking about. So if you have more than, we'll say 180 milligrams per deciliter of glucose in the blood, then where will you start to see glucose show up? In the urine. Okay, so you should not have glucose in your urine unless your blood glucose gets so high that all of your transporters are being used, you're over your Tmax, and we simply can't reabsorb it all anymore. We can't. So it's gonna stay in the fluid, it's gonna make its way all the way around, and it's gonna end up in the urine. We call that glycosuria, right? When there's glucose in the urine. And remember we said that glucose in the urine was associated with diabetes mellitus. Which diabetes mellitus, what's the hallmark there? Like what is diabetes? You have high blood sugar, right? So you have chronically high blood sugar, you're gonna chronically be going over your Tmax, so you're gonna chronically have glucose in the urine. And so glycosuria is really, really common in uncontrolled diabetes. The renal threshold for amino acids is a lot lower. It's only about 65 milligrams per deciliter. And it's really not that odd to have amino acids in your urine. It shouldn't be happening all the time. If you constantly have amino acids in your urine, that's a problem. But if you go to Outback and eat a big steak, you'll probably have amino acids in your urine later. It's a lot easier to overwhelm the Tmax for amino acids. We call that amino aciduria. Notice, this is not protein in the urine. Proteinuria is protein in the urine. That is a completely different monster. Okay, this is a single amino acid. That's small. That gets filtered out. It just means we couldn't reabsorb all of it. If you have actual proteins in the urine, that means you're forcing a very large substance across that filtration membrane somehow. So either the membrane's broken in some way, so there's some sort of kidney damage, or the pressure is so high that it's forcing really big things through the membrane. Okay, so really, really, really high pressures. You can have protein in the urine as well. All right, so we've got to spend a few slides talking about the actual pressures that are involved in glomerular filtration. Actually, this is a good point to